A very good afternoon to all. Now we have to see about the post fertilization changes in flower. First, we are observing an internal structure of one flower. Here, the from the outside, calyx, the greenish calyx, and the colorful petals, and the internal stamens and the pistils are displayed. Here, the stamens and pistils are only the reproductive parts. Those are only involving because of stamens are producing anthers and pollen grains, and the pistils are producing ovules. Both are fusing from the anthers. Pollen grains are transferred from the anther to the stigma and fusing with the ovule. That is the fertilization. Now, we already taught, we already studied that fertilization, the fusion of male gamete and female gamete, that thing we already studied. Now we have to study after the fertilization what is happening, what are the changes is happening in the plant, what are the organs are produced, new organs are produced or new organisms are produced, how, that thing we have to see. Okay, here you see after fertilization, first the greenish calyx that is also shed outside, and the colorful petals that is also degenerated. After that, after the fertilization, see here inside the ovary, male gamete and female gamete fused and formed the embryo. Okay, diploid embryo is formed. Okay, embryo should grow, embryo should germinate. For that, some endospermic tissue or endospermic food is also present inside that embryo or nearer with the embryo. Okay, then see in the pistil, many in the ovary, many seeds or ovules are converted into seed, and with the seed, while the seed is germinating, some two cotyledons are also formed. That is protective and food giving to largest leaf with the seed. Okay, that is also formed. After that, gradually the seed is germinating and the seed is liberated from the parent plant and liberated into a soil and producing a new plant and producing a new flower also. Okay, then the next one, here what are the organs? Here, the parts before the fertilization, what are the organs in the flower, and that part is converted into what? Listen, parts before the fertilization and the transformation after fertilization. Listen, the colorful greenish sepals and petals and the male parts of stamens and the female parts stylus and stigma, all the parts are usually wither and fall off. That is shedding from the flower. And the next one, ovary, the female pistil. At the bottom of the pistil, ovary is a sac like structure. Inside the ovary only ovule is present. That ovule is only giving seed and the ovary is only giving fruit. Okay, ovary is a sac like structure that is producing fruit. Inside the ovary, ovule is present, that is converted into seed. Then the next one, egg, the two gametes are fusing, that is formed the egg. The two gametes are fusing, that is only formed the egg. That egg is converted into a zygote. Zygote is the first cell of the next generation. Then the next one, funicle. Funicle is the stalk like structure, that is a stalk of a seed. Then the next one, micropyle. Micropyle is a small opening, minute opening of the ovule. That is also the minute opening of the seed also, through which only O2 and the water is going and coming from the seed and the outside of the seed. Then the next one, nucellus. Inside the ovule, nucellus tissue is present, that is converted into perisperm. And the outer integument, integument means covering layers of a ovule. The outer covering layer, that is outer integument, that is converted into outer seed coat, testa. The inner covering layer, that is a inner integument, that is converted into inner seed coat, that is a tegment. Then the next one, synergid cells with the egg cell, two extra synergid cells are present, that is also degenerated. And the secondary nucleus, see here at the bottom egg is present, and at the opposite end antipodals are also present. Here see the secondary nucleus at the middle of the embryo sac, secondary nucleus to secondary nucleus is also present. Okay, so that is converted into a secondary nucleus is converted into a endosperm, and the antipodal cells, the opposite end of the, at the bottom egg is present, and at the opposite end of the egg antipodals are also present. Here the antipodals also degenerating. Okay, now while fertilization, see, while fertilization, from the male gamete one nucleus is coming, and from the female thing one egg is coming, both are fusing and forming a 2n type of zygote is formed. And the next one, from except the first fertilization, from the second male gamete is also coming from the male gamete, male part, and the from the female part and the secondary nucleus that is also coming, that is having 2n or 2 nucleus. Here at the same time, three nucleus are involving, that is also fusing together, so that two times fertilization is happening, so that it is called the double fertilization. Here at the second fertilization, male gamete one nucleus, the female part, from the female part two nucleus is coming, so at the same time three nucleus are fusing together, so that it is called the triple fusion, and that is forming the endosperms. Okay, such a way only the zygote and the endosperms are forming. Now the zygote is converted into embryo, and the endosperm is giving the food to the growing embryo. Okay, now the embryo, how the embryo is developing or growing inside the seed, that thing we will see. Okay, here some exception cases are given. Listen, the receptacle becomes a fleshy and the edible around the fruit, enclosing the seeds as in Pyrus malus, apple. The calyx, the outer region of the flower, may persist and the enlarge. For an example, solana, brinjal, in which the calyx is present at the vegetable and the fruit stage also. Then the third one, the flower stalk or axis below the gynoecium enlarges into a juicy, pear-shaped body, which is edible anacardium. Then the last one, the cells present at the tip of the outer integument around the micropyle develop into a fleshy structure called caruncle. Okay, then the funicle, for each and every one, the diagram picture is also given. The funicle develops into a fleshy structure which is often very colorful and called aril. See the aril. Okay, like this, different some exception cases are also happening in the post fertilization changes. Okay, then now we have to study the endosperm. What is endosperm? Already we told endosperm is the endosperm that is a happening by the, that is formed by the triple fusion inside the ovule, the two secondary nucleus and one male gamete, all the things are fusing to form a endosperm. From the primary endosperm nucleus, by the process of triple fusion, endosperm is formed. At the top is it is given, and it should happen before the zygote starts to divide. The primary endosperm starts to divide to form a endosperm, but it should happen before zygote starts to develop. Okay, then the endosperm are of many types due to the different type of development. The first one is nuclear endosperm. Here how the endosperm is developed, due to that the endosperm was divided into 3 and the extra one type 4. Okay, nuclear endosperm, first one, see the diagram. Nuclear endosperm, in which the primary endosperm nucleus that is divided into 2, that is undergoing the mitosis cell division. Here continuous mitotic cell divisions form a many number of free nucleuses are formed. Here after forming the main nucleus, there is no cell wall formation is happening in that. Okay, so continuously mitotic cell division is happening, so that nucleus is only multipled and present freely in the endosperm. So there is no cell wall, so that there are no complete cells are also present. Inside that nucleus is only present, so that it is a nuclear endosperm. Example, coccinia, capsula and arachis. Then cellular endosperm, see the diagram. Cellular endosperm, here primary endosperm nucleus that is divided into 2 nuclei. Okay, here the two nuclei that is also undergoing the continuous mitotic cell division and forming instantly a cell wall formation also, so that here complete cells are also formed, so that it is called the cellular endosperm. Example, Adoxia, Helianthus and Scoparia. Okay, then next one, Helobial endosperm. See the diagram, Helobial endosperm. See here, Helobial endosperm. Here the primary endosperm nucleus as usual it divides, and nearer to the micropellar regions the cells are continuously doing the mitotic cell division, and many free nuclear substances are only present, and nearer to the chalazal end the nucleus is forming the cell wall, so that complete cells are formed near to the chalazal end. So at the one end cell wall formed cells are present, at the another end free nucleus containing cells are only free nuclei are only present, so that two types of cell forms are present. It is called the helobial endosperm. Example, Hydrilla and Vallisneria, that is a water living plant in which helobial endosperm type of endosperm is only present. Ruminant endosperm, last one, it is on the surface. You see the surface, a ruminate or hard or unevenly or rough at the surface, irregularly and unevenness in its surface. Example, passiflora and myristica. So in this not only its surface, the overall shape is irregular in shape, and the surface is also unevenness and the rough in nature. That is called the ruminant endosperm. Example, passiflora and myristica. Functions. The endosperm, the first very important function of the endosperm is the nutrition. It is only giving food to the growing embryo. So the main important function of the endosperm is giving food or nutritive function only. Okay, then process mode of embryo development, the accurate or clear or exact mode of embryo development is also happening by the endosperm. Okay, then next one, development of dicot. How the dicot embryo, here the development of dicot embryo should come. At first, listen, one embryo should develop into a complete plant. At first it should produce a zygote cell. Zygote is the first cell of the producing a whole plant, because of from which only a 2n type of sporophytic cell is formed. Okay, from the zygote terminal, the zygote cell is divided into is undergoing the mitotic cell division, horizontal division. First you see the diagram. First you see the zygote is divided into periclinial division. Periclinial means horizontal division, anticlinial means vertical division. Here it undergoes a periclinial division and two cells are formed at the upper terminal cell or upper cell, the base cell or lower cell. Two cells are formed. After that the upper cell is divided into two, the lower cell is also into two. Continuously the cells are divided into many cells, so that it undergoes a globular stage and the heart stage, heart shaped embryo, and the matured embryo. At the final stage, you see inside the embryo what are the things are present. See, that is a matured embryo. Matured embryo in a seed. Inside the seed, a matured embryo, how it is organized, what are the parts are also present inside that. See, cotyledons, that is embryonic leaves. Cotyledons, the embryonic leaves, that is also present. The upper part plumule, that is that leads to produce the shoot, and at the lower part radical tissue, that is producing root. Okay, and the root, nearer to the root cap is also present, and such that many parts are also present. In further slide we'll see. Okay, then seed. One embryo should grow and should develop into and produce a new plant. For this, inside the seed only the embryo should grow, so that what is seed, how it is produced, from where it is produced. Ovule inside the embryo sac, or inside that one full ovule, a complete ovule is converted into a seed. The next one, In the seed, embryo, that is a growing organ, growing part, then endosperm, that is a nutritive part, then protective outer coat. These three things are present. Then in the seed, if the nutritive content is more, that is a albuminous or endospermous seed. Example, wheat, maize, barley. But sometimes the nutritive content is less, or there is no nutritive part, that is a X albuminous or non-endospermous. Example, mango, bean, cucurbit. Okay, some seed size is also varies from in different plants. For an example, orchid seed 20.33 milligram, and the double coconut is having the 6 kg of tender coconut seed is also having. Then structure of dicot seed. See the first diagram. See the overall seed is overall dicot seed is given here. Caesar plant is taken. See overall dicot seed is elaborated, and the parts is also given here. Here a seed that is present inside the fruit is only. Okay, so seed is attached with the fruit wall or with the fruit by a stalk like region, that is called the funiculus. Okay, funiculus. Okay, the attaching marking is called the hilum. Okay, after removing that hilum, a small minute opening is called the micropyle, through which only water action is going, CO2 is coming, water is going and coming. The pathway, a minute opening is called the micropyle only. Okay, then surrounding the seed, the integuments, inside the ovule, surrounding the ovule, outer integument, inner integument. The outer integument is converted into a testa or outer coat of the seed. The inner integument is converted into a inner tegment or seed coat. Okay, so that the integuments are only converted into a seed coats, outer and inner seed coats. Okay, except that radicle. See the radicle tissue, radicle. Inside that, from the lower axis is having some radical tissue. From the radical tissue only root is coming, root is growing, or root is developed. And the upper part, see plumule, that is a tissue from which only shoot is developed. Okay, here another one thing, epicartile and hypocartile. Okay, above the level of cotyledons, the length of the axis is called the epicartile, and below the level of cotyledons, the length of the radical region or the length of the lower region is called the hypocartile. So these are the internal structure of a whole dicot seed. Okay, then structure of monocot seed. See monocot seed. For the monocot seed, we take the oryza plant. Here caryopsis, a single seeded simple outer coating, or there is no fleshy part. That type of seed is called the caryopsis. Here outer brownish husk or outer small brownish husk like regions, that is also present. Here inside the seed, what is present, what are the things are present, we will see. Listen, normally a seed containing endosperm or the nutritive tissue and the embryo, these two things are important. Here the two things are separated by the epithelium layer. Listen, the yellowish part, that is a growing embryo, and the thick upper part, that is the growing embryo. From the growing embryo, or the nutritive parts or nutritive content is taken to the growing embryo by the shield like, or by the outer coating, the shield outer coating, that is called epithelium. See here also given, through the epithelium only the food is coming. Okay, here some other things also given. See, listen, here the nutritive tissue, that is not a very important, because of that is giving the food only, that is not involved in some other functions. Okay, here the embryo, in which at the lower part, radicle. In dicot seed also the radicle is present. From the radical tissue only root is developed, and the root cap is also present in the monocot. Okay, the radicle is covered by the coleorhiza layer, and at the top, see, the plumule is present. That is covered with the coleoptile layer. Here the overall embryo The overall embryo is looking like a shield shaped, okay, and that is that lies towards the lateral side, okay. That is called, the overall embryo is called the scutellum, okay. The overall embryo, that is a shield shaped structure, that is called the scutellum, okay. Then the next one, apomixis, okay. The embryo development, all the things we saw. Now apomixis is what? Apomixis means the male gamete and female gamete is mixing, that is a mixis, okay. Ampemixis means, ampe means two, male gamete and female gamete is mixed, that is a ampemixis. Here apomixis is male gamete and female gamete is not mixing, that is a fertilization is not happening here. Before the fertilization, meiosis cell division should happen, that is also not happened in the apomixis, and at the same time both the gametes is are also not mixing, okay, not united, not fused. The two things are not happening here, that is a apomixis, okay. Winkler 1908 who discovered that apomixis, and in which the two things are present, recurrent and non-recurrent. Two type of apomixis is present, in which in the non-recurrent the meiosis can happen after forming the four megaspore mother cells. The megaspore mother cells is megaspore mother cells are normal megaspores that is not fusing with the male gamete. But here in the recurrent apomixis, here there is no meiosis and there is no mixing of male gamete and female gametes. That is a fertilization. Fertilization is also not there, and the meiosis is also not happening here. That is a recurrent. The recurrent apomixis, that is also divided into two. That is a vegetative reproduction, vegetative apomixis, and the agamospermy. In the vegetative reproduction or vegetative apomixis, vegetative reproduction means vegetative apomixis, in which by the runner, sucker, bulbil, and some bud, by these vegetative parts the embryo is formed. But in the agamospermy, another, that is divided into, that is classified into again three types. That is a adventitious embryony, diplospory, and apospory. Adventitious embryony, by the, we all know that ovule, that is in the ovule, outside of the embryo sac some nucellar tissues is present. That is a nucellar tissue means that is a 2n or sporophytic cells, okay. So from the nucellar tissues the embryo is formed, that is a adventitious embryony. Diplospory, diplospory is what? From the megaspore mother cell itself the embryo is formed, that is a diplospory. Because of that is a megaspore mother cell is that is not haploid, that is a diploid only. After happening the meiosis only it will become a haploid. Up to that it will be only meiotic condition. From the meiotic condition cell, that is a megaspore mother cell from which the embryo is formed, that is a diplospory. And the third one is apospory. Here the meiosis cell division is happening, so four megaspores are formed, but the four megaspores will be degenerated, and finally, like the first step of adventitious embryony, from the nucellar tissue only, the embryo is evolved, the embryo is coming, okay. And at the last, some polyembryony, cleavage polyembryony. Polyembryony means what? More than one embryo is formed at the same time. At one time, more than one embryo is formed, that is a polyembryony, in which the cleavage polyembryony, one embryo is cleaved into many. For an example, orchids. Then second one, formation of embryo by the cells of embryo sac other than egg, except the egg, from other cells. For an example, cyanogytes, cyanogytes near to the female egg, two cyanogytes are present. For an example, Aristolochia. From the cyanogytes also the embryo is coming. Then the next one, third one, development of more than one embryo sac. The first one, more than one embryo. The third one is more than one embryo sac, means that is a multiple embryo sac are formed, and each and every embryo sac is producing a new one or new plant. Then the fourth one is activation of some more sporophytic cells of the ovule, like a second one. Here diagram is also given. At the top, zygote is present. From the zygote, the embryo is formed, that is a zygotic embryo. At the bottom, Antipodals are present. From the antipodals regions also embryo is developed. So that is a antipodal embryo. Okay. Here also giving almus glabra showing the zygotic and antipodal embryo. Okay. So that is a here more than one embryo. Two embryos are present. Upper one, lower one. So this is a this is also a polyembryony. Okay. Then the next one, last topic. The last topic, parthenocarpy. Parthenocarpy means we all know that fertilization, the last topic, the last class topic, fertilization. Fertilization means the male gametes and the female egg both should fuse and producing a zygote. Okay, that is a fertilization process. Okay, here there is no fertilization. There should not be a fertilization. Without fertilization, the fruits is formed from the egg or from the parts of the female part. That is called the parthenocarpy. Example, seedless fruits, for an example, grapes, banana, and many commercial fruits are produced now. In many places we are seeing seedless fruits. Many fleshy fruits are we are buying. That is a parthenocarpic fruits. Niche 1963 only classified the parthenocarpy, for an example, the genetic parthenocarpy. In genetic parthenocarpy, what is the process is happening? Hybridization or mutation. Hybridization means two different individuals, two different varieties are fused or crossed to produce a new one. Its gene is converted into another one. In mutation also, citrus plant, for an example, mutation also, sudden heritable change. Suddenly some heritable changes, heritable means the DNA changes, that will change the overall plant characters also. Okay, it will also help us to produce a commercial usages. Okay, then the environmental parthenocarpy, frost, fog, temperature, low or high temperature, we are artificially giving these environmental conditions. The parthenocarpy can happen. For an example, low temperature, nearly 16 hours here given, but 3 to 19 hours only. If we give the low temperature for 19 hours in pures, the parthenocarpy can happen. Then chemical induced parthenocarpy, gibberellin, GIBB, not PPBB, gibberellins and axins. That is a growth regulators. If you apply that gibberellin axins also, the parthenocarpy can happen. Okay. Then applications in the horticulture, in the horticulture, more products we can get, and for the commercial purposes. In the commercial purposes and in the trade, many seedless fruits, if we give the or if we sell the seedless fruits only, we can sell the more products, more of our products, then more edible portion. In parthenocarpic fruits, the seeds are not there, so that we are if we see the seeds, we should take and we should remove one by one, but in the more edible parts is there, we can get more and more, so that is also important one. Then jam, chaws, these are then many other purposes. For many other purposes, we are using the parthenocarpic fruits artificially. Okay, these are the post fertilization changes. In this lesson, first we taught we studied that the asexual reproduction in plants, then the sexual reproduction in which fertilization and pollination. First process should happen the pollination only. The male pollen grains that should travel from the anther to the stigma, it should come there. That travel is called the pollination. After that, fertilization. Now after the fertilization, what are the processes will happen? That things we saw in the class. Okay. Okay, now we will see in the next lesson. Thank you.